So presenting myself, I'm Thomas Cole. I'm a co-developer of the resilience design sort of toolkit. I'm a trainer in RD, and I advise philanthropic organizations. Um, we have Warren Brush, who's also a co-developer of RD and sort of a global teacher and consultant implementer. He's an applied ecologist. And we have David Kalama, who's, I call him the professor. He's the research technician and science liaison officer with African Women Rising, coming out of Gulu University, but he's based out of our Loch Neno Training Center in Palabek Refugee Settlement. So the three of us are talking today. Next. So what is RD? So, what makes it different? So it's an ecologically based approach to regenerative agriculture. And it was developed specifically looking at the type of programs, humanitarian and development, that many of us are running. They're really unique sort of operating environments, both in terms of climate, in terms of donors, and this and that. So it's building on agroecology, permaculture, climate smart practices, conservation agriculture. So it blends a lot of these different pieces together. But the four core areas is whole systems design. It uses a whole systems design approach, and we look at stabilizing water, so hydrological stabilization, creating living soils, and so that's what this session is today, and building biodiverse systems. So those four core pieces. And it's a practical approach that empowers farmers, it empowers extension workers, it empowers heads of organizations, managers to understand how to observe and implement and adapt their programs to fit very context-specific situations. And since it's a principle-based approach, it can be worked at the household in very small gardens, all the way through large field crop gardens to community stabilization, watershed approaches, all the way up to sort of community and broader watersheds. And the goals are food security, income, building out ecosystem services, and building self-reliance in farmers. As I said, you can learn a lot more at the website globalresiliencedesign.com, but we're going to get into really the nuts and bolts right now of the soils piece.
Thank you, Tom, for bringing us into talking about this subject. And hello, everyone. It's an honor to be here with you. And yeah, I want to hopefully answer this question through this next series of slides. Next, Tobias. And I'm thinking about how often our programming, especially our agronomic programming, focuses on the chemistry uh, and and not necessarily looking so deeply into the biological functionality that is a part of a natural soil system. And what my hope is by the end of this presentation, you'll have a good idea and a good understanding of what not only what the outcomes of a good soil biological system is, but also how to get there. How do you develop it? And then how do you monitor it and look at that, which is what David Kalama will bring into it. So when we step back and we start to look at the functions of the soil biology, there are many, and we're going to get into more and more of what those are as we get into this presentation. But what I'd like to do is I'd like to actually step back and look at the bigger patterns around the effects of soil biology on human health. And go ahead next, T. And as we step back and take like a bird's eye view of the soils and the impacts on human health, some of the recent research that's been coming out around what we call hidden hunger, which is when we have calories in our food, but the micronutrients are missing. And we'll talk a little bit about why this is really important and how it relates to the soil biology. Because right now there are billions of people who are suffering from hidden hunger, which is that deficiency in micronutrients. And really, there's not a calorie shortage, but there's this nutrient collapse that has to do with micronutrients. Next, T. And so when we start to look at food nutrient density in the plants that we're growing that are feeding ourselves, that are feeding the people that we're working with, what we're seeing is that the declines in mineral, vitamin, and also in other types of nutrient content that's needed for health. So things like protein density, the loss of calcium, magnesium, the loss of phytonutrients, and then, of course, the decline in antioxidant capacity, which is also directly translates to our capacity to stay healthy. And so it's interesting in next T, when you start to look at what that means for our climate resilience, I think it's a really important issue to look at plants' capacity to be climate resilient. A singular plant, we can help to make climate resilient. And there's a lot of research out there that shows how plants are, when they have a lot of micronutrient density within them, that they are, and macronutrient density within them, that they have the ability to withstand climate shocks. And that being, I know many of you are probably a part of a climate program, or your title has the word climate in it, or has the word resilience in it. And yet, so often our programming doesn't look at actually how we're creating and programming climate resilience into the very plants that provide the base foundation of nutrition and also of income. Next, Steve. So, this is an interesting one. When we start to look at what are the types of things that we're doing in our programs that may be causing the collapse of soil biology, and looking at how the types of agronomic practices that we use have followed a pathway of the collapse of the nutrient decline that we're seeing. So some of the things that we see that cause the soil microbial life to decline is repeated tillage, where we're breaking up the soil aggregate stability of that soil, because so much of the soil biology is needed to actually have good soil structure. We start to see that we're using synthetic nitrogen reliance, reduction of organic matter. In a natural system with a healthy soil biome, we're cycling organic matter naturally, and we'll talk about that quite extensively here in a few minutes. We see that there's a loss of soil cover. So we're having clean fields where the soils are not covered. They're not with either living or dead mulches. There's a lot of compaction. All the ability of the soils to respire and to breathe, to be aerobic, which is such an important part of having a healthy plant that has the ability to get the micronutrients it needs. It needs to be growing from a aerobic environment. And so when we collapse and compact the soils, we create more of an anaerobic environment, which is where pest and disease start to take over. Fungicide use also kills the beneficial fungi, and we'll talk more about what fungi can do for our systems. And then, of course, having monocultures have replaced so much of our polyculture systems. And those of you who have been through a resilience design training will learn how incredible biodiversity and using annuals and perennials in a system creates one of the best soil environments possible. And then, of course, the disrupted carbon cycles. Next. So, soil microbes control a lot of processes for life, just life in general, because if we're eating plants, our animals are eating plants, then that translates to us as well. So things that soil microbes control is mineralization. So they are are amazing at unlocking the minerals and converting them into plant soluble forms, into usable forms. This process of chelation, so keeping micronutrients soluble and mobile in the root zone. There's also the mycorrhizal, which is that beneficial fungi that I was just talking about, and how that mycorrhizal root net, if it's well developed, can actually go beyond the roots of the plants themselves to gather more nutrients for those plants. So you're actually extending the feeding capacity of those plants by having a healthy mycorrhizal network. You're also creating nitrogen fixation without using synthetic fertilizers. And there's a lot of ways that the soil microbes help with that and also translate that nitrogen from the atmosphere, which is so prevalent and available, and it helps to convert it into plant soluble nitrogen that the plants need as one of the many nutrients that they need to be healthy. There's this whole process of biocycling that's important. So where the transferring of nutrients between layers. So if you have mulches on the top that are diverse and are and protected of the soils, that the nutrients from that can actually be drawn down through the different layers of the soil and being cycled through. And we'll talk more about that in a minute. Plant immune signaling as well as soil aggregation. So you have with bacterial gels, you have this ability to create in the soils really healthy soil structure that holds the aggregates together. Next, T. So one of the latest studies that has come out, well, it's not it's in the last like 15 years. There was a study that showed that that when soil microbes decline, plants start to take up more water and carbohydrate mass, but that there's fewer minerals, vitamins, and phytonutrients. Thus, the yield rises, but the nutrition falls. And I think this is a really important thing to understand is that in humanitarian programming, we don't want to promote a nutrient dilution effect where we get high yield but low nourishment. I know those high yield pictures you show your donors are the ones you want to, you want to show that field of maize really producing. But what we really need to do is look into how well established is that plant with its micronutrients, because that's going to feed somebody and help them to be more disease free and to be healthier in their development. Or our animals that are eating those plants will also be healthier. And so it's an important thing to think of when we start to look at programming. Are we programming nutrient diluted plants or nutrient dense plants? And at the end of this presentation, David Kalama will walk us through some of the ways that we can measure that. Next. So hidden hunger and soil decline go together. And so if soils lose their nutrient density, crops lose their nutrient density. Now it's really important to understand that. And when we say lose their nutrient density in the soils, that directly translates to soil biology. And if that soil biology is sick, it starts to translate to the food itself. And then if crops lose their nutrient density, diets in the people and our animals lose their nutrient density. And if diets lose nutrient density, the whole population of people and animals starts to move into a more diminished micronutrient makeup in our bodies, which then translates to more disease and pests in our life. And so it really the soil biology is a direct translation to how our human health is actually being maintained or promoted and built, or is it in decline? Next, T. So let's get into what is soil biology. Now that we've talked about kind of that bigger picture, and let me walk you through some of the basics of what soil biology is for those of you who haven't maybe been exposed to this before. Next, T. So a healthy soil biology is a really it's an ecosystem amongst itself in the soils, and it's an aerobic ecosystem. When we again, as I mentioned, if we're in an anaerobic situation where we've choked out, we've compacted the soils, we've made it so that oxygen and other respiration from the atmosphere, atmospheric respiration doesn't happen in the soils, you start to get that anaerobic, which is where we start to get the fungal diseases. We start to get a lot of other the plants start to get weaker. You have less translation of micronutrients in that as well. So then, it's amazing in a healthy soil food web. There are lots of different trophic layers and that's the next slide, T. There's a lot of different trophic layers of um, ecology happening where at the very base of it, and this is where it's really important, is the fungi and bacteria. So we're looking at the relationship and the balance between the fungi and bacteria. So often in an anaerobic soil, you'll get types of bacteria that are more anaerobic bacteria. So the type of, when you smell soils that are stinky and they don't smell good, they smell sour, that's when you're getting that bacteria that is the anaerobic, which is telling you those soils are actually going to cause harm to your plants. It's when you start to get striga in the maize when you're growing it. And so that it's important that we start with the fungi and bacteria that are actually aerobic. Next, T. And so at the foundation, bacteria are primary decomposers. So those of us who are talking about organic matter and the need for organic matter in our soils, we have to also be talking about the systems that can actually digest and convert that organic matter into plant soluble nutrients. And bacteria are one of those primary decomposers that do that, as well as they produce this gel that helps to hold the aggregate soil materials in a way that gives us good soil structure. So your soil structure collapses if you kill that bacteria with chemicals. So the thing that I think is really interesting with this and with the fungi, next T, is that the bacteria will decompose a lot of materials like the basic organic materials, but some of the more complex matter is broken down. So if you have like um, fish waste, or you have animal waste or manures, the fungi helps to break that down and to create more of a plant soluble nutrient. They also help to transport nutrients right to the root zones. And so you get some of the fungi will either be ectomycorrhizal or endomycorrhizal, meaning that they'll either surround the root zone of the plant, or they'll actually penetrate into it and they'll share nutrients. And then the plants will also give sugars to the fungi in exchange for the nutrients that it brings. One of the things that happens in the whole trophic level of eating that happens in the soil zone with all the soil biology is that then you get nematodes and you get other soil organisms that are actually giving they are eating the bacteria and the fungi. And what that's doing is then they are manuring out the nutrient from them. So they're digesting it, and then they're the higher level of the trophic layer. So the kind of the protozoa, the nematodes, even some of the different mites and things that are there will eat. You'll start to see them eating each other, and then they manure out the plant soluble nutrient. Next, T. And then as you start to step back in looking at what is the most cost-effective way to end hunger and create climate resilience crops, what we can see is that it's actually developing and restoring the soil biology and building it. And so the way we work in resilience design, we're focused on the soil biology as one of the core components of how we're actually developing our systems. Next, T. So why are they important? This is really important. Why then these living soils? We understand that they're there if we're tending to them, and we'll talk about how to do that after this. But this is what's really important: is they build soil structure that if you have a healthy soil biology, disease has nowhere to grow. All the microsites on the plant are on the in the rhizosphere in the root zones are covered with beneficial bacteria and fungi. The life in the soil also eats insect pests as eggs and larvae. So it ends up that the insect pests that we usually see that cause damage, a lot of the leaf eaters that we see on the plants that grow from eggs and larvae in the soils, they get eaten by the healthy soil biome and actually become nutrient that feeds the plants rather than something that destroys the plants. They retain nutrients in the soil and they keep them from leaching out, which is so much of our soils right now people put NPK in them and that so much of it is leached out and it's less soluble for the plants to use them as well. They convert nutrients into forms that plants can use, as well as they make the nutrients available when the plants need them. It's like if you are wanting, you're craving certain foods because your body needs foods, and you eat that at the time of your craving, you're feeding it when your body's needing it. And plants are the same way. And yet, so often they don't have the nutrients available to them when they need them, when their structure or their growing in their point of growth that they need different types of nutrients, they're not available. They also suppress weeds, which I think is interesting. And there's a lot of reasons behind that. Next, T. They increase water holding capacity. Those of us who are looking at how do you create a drought resilient growing system, having the capacity of the soils to actually retain water and also limit what is the usage needs of that plant or that crop by having the soil biology in a healthy state. So that one of the things we often talk about in our permagarden program is how the way we design it is that it's a garden on top of a water tank. And so we've created a garden basically atop a soil system where that soil system is healthy and aerobic and has a lot of porousness to it to receive the rain so that it can then be held for a time when the rains are not there. We also are seeing, and a lot of the research shows us that the healthy soil biome helps us to extend our growing seasons. So we're getting more and more bridge crops from the rainy season into the drier seasons. It also increases rooting depth, which gives you the capacity to have more biointensive planting because the plants can spread their roots deeper rather than laterally. And then it increases generally to profits and family nutrition. Next, T. So, soil biology, nutrient density, and pest and disease resistance. I've talked a little bit about this, but again, I just wanted to reiterate. Oftentimes we hear people say, "Oh, yeah, I have a pest problem." And what I want to relate back to this talk is that no, you have a soil biology problem, that you actually have a weak plant. So, the pest and disease are a symptom of something in the system not working. And so often it is the soil biome has been destroyed. The conditions of the soil to be aerobic versus anaerobic are have been created by poor agronomic practices. And so when we start to really look at this, how then do we design so that the soil biology is present, is healthy, and can help us with pest and disease resistance. Next slide, T. And so how do we create living soils in gardens and farms? Next slide. And I'm going to walk you through 10 different key areas that are important to develop a healthy soil biome. Next, T. So deep soil preparation with organic matter or organic material. So oftentimes we do double digging, or on larger farms we do what's called single digging, where we preserve the existing topsoil and then we break up the subsoil. So just think about that for a minute. You're digging deeper so that you're creating more of a root zone for the plants that you're planting. But in the subsoil, you're actually integrating a lot of different organic materials. Like in some of the programs with African Women Rising, we're putting up to 15 different types of organic materials, everything from different types of manures, different types of leaves and grasses, bone meals, blood meals, all these different things are going into the subsoils. And then we put the topsoil back on, and now we have up to 60 centimeters of free-flowing soil with deep soil amendment that the roots can go down and create deep tap roots for those plants to be healthy from the start. And that deep soil preparation allows us to grow more intensively. And we've seen up to 20 or 30% more plants could be in the same area. It's like you've expanded your farm 20 to 30% by doing this particular process. So just keep that in mind. Deep soil preparation with organic material, many diverse foods to be converted into different types of micronutrients. The diversity of input is just as important as the diversity of mulches, as the diversity of plants growing in those beds. Next one, T. Just a small increase in soil organic matter significantly increases the nutrient density in crops. And so that just talks about that first slide. So high soil organic matter and richer microbial communities correlate with higher micronutrient and phytochemical content in the crops. So healthy soil biology produces nutrient dense foods. Next slide, T. So diversity of organic matter, I talked a little bit about that, and then also mineral input. So there's the double digging, but then how we embed organic materials into it is very important. We also, in the topsoils, we do a mix of dried compost and dried manures to be able to have some more nutrients near the topsoil as well. But the diversity is really important. And one of the we have a resource that I think Abby will put in at the end here into the chat, which is a breakdown of all these different nutrients that we put, or amendments we put in the soil, and what their nutrient contents is. Another way that we help to develop the soil biology is we'll inoculate our subsoils with some of the local forest leaf litter, or a more developed agricultural systems leaf litter that has the biology in it. So we're inoculating as well. Next, T. Biodiverse and biointensive planting and growing beds. So biodiversity is an important part of maintaining the soil biology and what's underground. When you get a monocrop, it starts to limit the diversity that happens underground. So whatever you're doing above ground also happens below ground. So it's important for us to understand what are the companion plantings that we do. When you look at, and I think we're actually going to do a learning series about companion planting, or what we call guilds, which are plants that like to grow together. Like maize does not like to grow alone, and it's less healthy and less nutrient dense when it does grow alone. And there's a lot of research around that. Next, T. Protection from overgrazing and human footsteps. So having well-defined pathways, fencing, being able to protect from that compaction from overgrazing and human footsteps. Number 5, please, or next one, T. Living in dry mulches and diversity of mulches. That's very important. You don't want to use just a single type of mulch, but having many types of mulches is much better because the mulches themselves become food for the soil biology, and it actually becomes the next season's amendment, and the soil biology will pull it down into the soils. It also helps to protect the soil environment from intense sun, from rainfall, and gives a good insulation layer. Next, growing without chemicals. I've been a commercial farmer for many years and never once used chemicals in a very profitable and large commercial system. There's no need for chemicals. In fact, it's the inorganic chemicals destroys the soil ecology and structure by killing the biology in the soils. We just there's a link that you can go onto our website, Global Resilience Design. Maybe Abby has already put that in there, but we did a whole talk on biofertilization, which is another strategy we use. Using only organic methods from local available organic materials or resources. We also do pest repellents that are made from organic materials as well. Next, T. And then protection from compaction, even from rain, but even from flood irrigation. Sometimes we do flood irrigation systems that compact the soils, like you see here. This is a system that is in Somalia where they were flooding from the river, but you can see that it was causing compaction. And so looking at how you can redesign the system to do lateral irrigation into the root zones. And that's another strategy that we'll use is not top flooding the growing system to cause compaction. And again, when we cause compaction, like I said earlier, we start to create anaerobic environments, which you can see these plants are struggling growing in an anaerobic environment with hot soils. And a lot here, you see a lot of evaporation happening as well. So you see desiccation in the plants because of the poor soil environment. Next, T. And then we see soil temperatures. We need to shade. We need to limit that the evaporation that happens at the soil level. So using just very simple shade structures or planted strategic shade, maybe on the west side of a growing area, you have some shade trees. As long as a plant, even in the rainy seasons, gets 5 hours of sun exposure, it will do fine and it will actually thrive. And so having west shade to help protect from intense, even in the rainy season, you get intense afternoon sun that can cause desiccation in the soils as well. So protecting from that. So shade from intense sun exposure. Next one, T. And then when no tillage. So we talk about basically doing we do soil disturbance in the very beginning, setting up the structure of the soil. Like I said, the double digging or even single digging. This is a wonderful trial that was done out in Isipe in Kenya. When I took this picture, this was two side-by-side systems, one with the typical tillage system on the right in the foreground, and then the background was a no tillage site. So that was a site that had been not tilled for 12 years. So this idea that you have to till what that does is it breaks up the aggregate stability of the surface soils and causes it to collapse, which then chokes the pores of the gaseous exchanges, and you start to see that the system doesn't produce as well. And so Asipe has some wonderful data on this as well. So this idea of no tillage, even on the largest farms in America that are doing no tillage, they're using machinery that does seed drilling. within the mulches. So this idea of we need to till is not true. It just is. It destroys the soil biology every time you do that. There are certain types of non-inversion plows that can help to open up landscapes without turning the soil to in and to help establish some vegetation and root systems that will help that soil to maintain good quality and good structure over time. Those types, there's one, there's a type of non-inversion plow called a keyline plow that we've used in Kenya and in other countries around the world. But often we're working with smallholder farmers, so we're basically just doing single dug, simple systems that are very easy to do and not much more work than if you were tillaging. That sets it up, and then from that point forward, if you take care of the soil, you don't have to dig again. And that's the beauty of it, is that you're not digging over and over. You're top dressing with good nutrients to keep that system fed so that the biology develops, so that every year your growing gets better, not worse. Next one, T. And then favoring locally adapted seeds that co-evolved with the soil microbes in that area, as well as the climate, the microclimates in that area. What we look for, this idea that you get certified seeds that were developed far away that have a terminator gene in them. We do not do that in any of our resilience design systems. We do not believe in that whatsoever. We realize that to have nutrition security and food security, you have to have open pollinated seeds that the farmers can locally adapt to with the soil biology. And that's a really important, the science is very clear about this. And so it's an important part of thinking of how do you create a good system of soil biology. You have to evolve it with the seeds that are being saved. Next, T. So I'm going to end on this slide before I hand it over to David Kalama to talk a little bit about how you monitor this. And basically, I just want to kind of recap that healthy soils are the foundation. And I should say healthy, biologically active living soils are the foundation of productive, nourishing, and climate resilient communities, because that's what happens with good healthy plants. You get climate resilient communities. And then by rebuilding the living soil ecosystem from the bacteria to the fungi, so building it from the base all the way through the beneficial insects and then the practices that we use, we can regenerate a lot of those natural cycles that feed us and our people. So really the key takeaway here is that when we design for life below ground, everything above ground begins to thrive. And so I'm going to hand this over to David Kalama. Hopefully, there was some good information in here for all of you, and we look forward to the question and answer as well. Thank you. David?
All right, thank you, everyone. Thank you, Warren and Thomas, for taking the lead in the early slides. I'll take over from here, from the slides where they're saying that why. So how do we measure soil biology in the field? Thank you, Warren, for the rich details about soil biology. And I'd love to take the members through the why and how do we need, or why do we need to measure soil biology in the field. Yeah, the why here is that we need to understand soil biology activity. As Warren has been explaining, all the conversions that happen within the soil is because of these microorganisms that are found. So you need to know the activity, and knowing the activity of the biological science behind the soil helps you also to understand how healthy the soil is and how productive the soil is. Then also guide decisions on soil amendments. You need to know your soil microbes. If you have like low fungal populations, then you need to do the required amendments that can help to boost particular microorganism that is lacking within the soil, which is very important for soil health and soil improvements. Then also guiding amendments or adding amendments to the soil. As for us, the R&D students or the R&D practitioners, we know that it improves crop resilience and productivity. So a moment you do amendments within the soil, you are supplementing what is lacking, or you are enriching, or you are adding more percentage onto the soil. So to make soil more resilient and also make crop more resilient and also productivity goes high and higher. Then it's also one of the most tracking tool for us to use. If we measure soil biology, it helps us to know how are we progressing in regeneration processes. For example, if you're RD practitioners, measuring soil biology is one of the way you can track how your program is progressing, how you are doing with your practices within the soil and also humanitarian area. Next. We need to know how do we know, maybe before any introduction of instrumentation, how do we know that our soil is good? Because as a farmer, as an extraction expert, or as someone building healthy soil, you need to have a preliminary understanding of what your soil should be. Actually, if you are regenerating the soil, you need to know where should I reach? And these are some of the key indicators that help you to know if your soil is well and if your soil is doing or performing well or is more resilient. And one of them is plant germination and growth is optimal. So if you see that your plants are doing well in the field, and maybe germination from one day and some days they are doing well, it shows that your soil is good and your soil is performing what is supposed to be done. And also it contains the required microbes that can support plant growth. Then also pest infestations and disease are minimal. Warren has just spoken about pest and disease infestation. They are just signs and symptoms that something is wrong. So if you have a lot of this, it means that your soil is not good. But if you have limited or little pest infestation and disease interference in your field, it shows that your soil is functioning well and also is doing the required work to be done. Then soils are porous, darker in color, smell rich with organic matter, crumble between your fingers. So these are some of the tests that you can move around in your field and try to judge your field. For example, if you pour some water, you can see that, yeah, the way it sinks with the water is okay. Water can move gently into the soil. It means that the percolation is fine. And also darker in color. We always know that a darker soil is always a good one for our productivity. Then the smell. Warren has been talking about the smell. I think it's one of the factors or one of the signs that will show you that organic matter is sufficient in your soil and means that soil health is good. And the smell should really be good. And I think a good soil has a very good smell. It actually attracts you as a farmer in the field. Then crumble between your fingers. So these are some tests that you can do in the field. Your soil particles should be able to roll within your palm. If you try to squeeze it, and should not really break easily or fall apart if you try to crumble around your fingers. So those are also some signs that can show you that your soil is well regenerated or is in good condition. It's also uncompacted. Uncompacted soil is poor. It's always prone to flooding and also poor infiltration of water within the soil aerations. So your soil should not be really so compact. If you try to dig out, it should really behave in a more porous way. And the soil lumps, if you pick in your hand, should really weigh in a very porous manner. So uncompacted soil is one of the signs. Then soil is protected by mulch and appropriate shade. Yeah, from my finding at Lockneno here, where we're trying to do a lot of experimentations, we see that most soils under kind of shading could be mulching or even native free zones. They perform well in terms of maintaining moisture and also soil microbial behaviors. So it means that giving appropriate shading to your soil will protect its health beyond what you may think. So if soil has good shading, it is also a sign that your soil is performing well and it's able to retain all the conditions required for productivity. Then higher bricks reading and microscopy. This one we're going to look at it in the next slide. We shall see that a better bricks reading reflects a good soil and also reflect good plant density, good nutrient density in the plants, as we're going to look under refractometer. And field testing. So this is one of the tests that you can also do. You can carry your equipment from home and you go try to check on your soil characteristics and properties in the field where your eyes cannot see. This one now track what you cannot see. So field testing is also one of the key areas that you can look at. We have simple and also complex field testing devices that we're going to look at. I think next. Yes, please. So here we have ways to measure soil biology in the field. Now, you can see that I have highlighted 2 points. Those are the 2 points that we are really working with. We are doing microbial biomass analysis and also refractometer at our demonstration site. But we have a list that are not limited to what we are presenting here, but there are so many that you can also explore within your time. So we have one of them is called soil respiration. Here we look at the soil microbiology activity in the soil. And as they behave in the soil, they always release gas, which is carbon dioxide. So one of the ways that you can measure soil biology is by trapping those gas, and you measure what quantity of carbon dioxide is able to be released within a specific period of time, given certain quantity of the soil samples. So you can use this in the field as well. We have apparatus like Solvita carbon dioxide burst that you can use directly in the field. Then also you can use a tight glass jar in the laboratory. You can take your soil sample in the laboratory and you add some water in the glass jar, then you try to trap carbon dioxide that is released from that soil sample. That gives you also the behavior of soil microbiology in the soil. Then also a good one for research is called infrared carbon dioxide analysis sensor. This one you can install and it keeps monitoring for you for a period of time where you will need to monitor your soil biological behavior. Then we also have another one called microbial biomass. One of these we are using is called microbiometer. We are going to look at it in details, but other techniques also under microbial biomass analysis include chloroform fumigation, which is one of the tools you can use to measure the biomass plus microbial community in the soil. Then also we have ATP assay. This is using energy compounds in the soil to estimate living biomass. It's quite technical. You can also explore when you get some time. And also we have under microbial biomass, we also have phospholipid fatty acid analysis. Yeah, these are all laboratory analysis, which is within your reach. We can try and see what one works for you. But for us, we have chosen using microbiometer as our simple field tool that can help us to analyze biomass plus microbial community within the soil sample we are regenerating. Then one of the complex one is also called DNA sequencing. This is more of genetics analysis. So this one, it helps you to classify microbiome profile within your soil. Here is more specific. It can help you to analyze how much or dominance is the bacteria and archaea in the soil, and also plus the fungi. They're using the ITS method, and the 16S is for bacteria majorly, and ITS is for fungi. This one helps to populate for you how much of those microbiomes you have in the soil. So it's also a very interesting tool to explore. Then soil organic matter test. You know, having good organic matter implies having good soil living organisms. So you can also use organic matter tests. One of it you can use Lawson ignition using a simple furnace, and you burn beyond the burning point of organic matter. Then the remaining you can calculate what was the organic matter contents in the soil. Then one of our key measurement here is also refractometer analysis. So this one helps us to measure plant density, which directly translates to what we call soil biology. I think in the next slide we shall look at that next. If I pick on the one we are loving most, that is using microbiometer. And here, what does it measure? When you're using microbiometer, you have to have in mind that you're going to measure microbial biomass in the soil. And this is the quantity of living microbes. Here, we are quantifying fungi plus bacteria in the soil. Now, it's reported in terms of micrograms, microbial carbon per gram of soil. So this is how you're going to be measuring microbial biomass using this microbiometer test kits, or a biomass index. You can call it a biomass index, or microbial carbon per gram of soil. Then it also helps you to figure out the fungal to bacterial ratio. It gives you directly what ratio of your soil sample contain bacteria and also contain fungi. So it indicates the balance between fungal and bacterial population. And here we're saying that fungal ratio is important because the higher fungal presence, the higher soils, the healthier is the soil, and also the better carbon storage and better aggregation for productivity. And also, if you have lower fungal population, it means that your soil is degraded or disturbed. It could be you are tilling it a lot, or you're using a lot of chemical in heavy farming systems. So those are some of the way we can judge from fungal to bacterial ratio. If our soil is healthy, or is moderately okay, or is very low. So it is good for us who are in regeneration practices to use such a tool to help us show our indicators. Next. Now, how do you use microbiometer? If you're measuring biomass in the soil plus microbial community composition. Now, this is a very easy step that I try to compile, and I summarize in both the photo and the text. Now, if number one, you're saying prepare your kit and labels. So you need to make sure that you have this kit. The photo is at the left-hand corner, upper side. So that's the kit that contains everything for microbiometer analysis. So you prepare your kit, then too you collect soil sample. Here we need soil from a zone of 0 to 15 centimeters. This is where
David?
most of the plant. Yes, please.
Sorry to interrupt you. You have about 5 minutes. So maybe
Yes,
we
please.
skip the steps. We can share
Yeah.
this with the presentation when we send it around to all participants. But I'm sure they would love to hear a bit about the refractometer as well. Thank you.
Okay, great. Thank you for that. We said to collect soil sample from 0 to 15 centimeters. This is where most of the activity takes place. So you need to, and most of the growth for our plant, the tender plants take place. So it's the desired soil level or soil depth. Then add extraction. So you can mix and thoroughly shake on your mixture. Then you can transfer the sample ready to your reading card. And in this, we are going to use a app called microbiometer, where you can figure out the reading of your fungal to bacteria ratio from, and also biomass details. Then the next slide, it is going to show us how you interpret the result from this kind of reading. Yes, next slide. Yes. So if you take your reading from the app, this is what you're going to get. And any reading that you get, you can refer to this summary. So if you have microbial biomass less than 200 micrograms per gram, it means that you have poor soil biology. So this is how you can interpret from 200 to 500. It is mean that it's an improving soil. The health is not bad, is in the medium status, and also greater than 500 is mean that your soil is healthy and is very productive. So this is how we use microbiometer results to interpret how our soil is. Then this is how we can also use the ratio. If the ratio of fungi to ratio to bacteria is less than 0.1, it means that the soil is still degraded and needs more effort to make it alive. Then balance is between 0.1 to 0.5, which is good for vegetation, and also high is from 0.5. If the ratio is from 0.5, it means that your soil is now better for to accommodate most of the plants. Yeah, next. Yes. Then another method that we are using also at our demonstration site is refractometer. Now, with the refractometer, it's a very simple tool that you can use and get your result within not more than 5 minutes, you have your result. So this one measures Brix, which represents the sugar and nutrient density of plant sap. So in this, we look at plant health, soil nutrient balance, microbial symbiosis, photosynthetic efficiency as our indicator for Brix findings. Now, how do we look at that? I think our next slide will give us more details of that. Next. Yes. Why Brix matters? Yeah, we can look at our indicators. Like if you have high Brix, it means that you have high nutrient density and stronger immunity. If you are measuring your plant, this is high Brix, meaning it means that the plant has high nutrient density and also have a strong immunity. Then low Brix, it means that the plant is stressed. Could be for many reasons. Maybe one of them is soil biology and poor soil biology, as mentioned in the slide. It helps farmers adjust nutrition instantly. For example, foliar sprays, compost teas, irrigation changes, and so forth. So this is how bricks can help you to make decisions among your regeneration processes. Next slide. And we see how we can use refractometer to measure this bricks reading. It's a simple tools that we can use. Then this is a 4 or 5 steps. You can collect fresh leaf or stem sap or fruit juice. These are majorly the tools that you're going to use: fresh leaf, stem, sap, or fruit juice. Then place 1 to 2 drops of refractometer glass on the refractometer glass. Close the lid and point towards the light. So this is the photo of the refractometer. Then at the lower in the circle is how the reading looks like. If you put your eyes in the refractometer, this is what you're going to see. And you're saying read the bricks value on the scale, then clean the refractometer for accuracy. So you can repeat as many times as possible with the same sample to confirm your reading. And this is how you're going to interpret in the next slide. For example, in this reading is saying 10, approximately 10 or slightly above 10. What does it translate to? So we have scientists who have developed the index reading for most of the crops that we have. So this is a sample table that I picked to help us understand the reading from the bricks. For example, we have tomatoes. If you read through the refractometer and it's giving you less than 6 bricks, it means that it is low in nutrients. Then the nutrient density in that tomato is low. Then if it is medium, it is between 6 and 8. And greater than 8 is a perfect one. It's an excellent fruit if you're testing for tomatoes. And it goes for all sorts of plant that we grow around us, and all the products have their readings. So probably I think we can have access to this table somewhere as our tools to use. Next. So as I conclude, how do we link the bricks reading to soil microbiomes and probably to you, human? So we have high microbial biomass leads to a better nutrient cycling. So if you have good population microbiology, it means that nutrient cycling is perfect, and having better nutrient cycling lead to higher bricks reading. And we say that higher bricks reading means that we have high nutrient density within the plants. And higher bricks reading means that the plant is stronger in terms of immunity. And having a stronger plant immunity means that there's a high possibility of having high yields and resilience of the plant wherever you're producing the plant from. And as I conclude, higher yields with better nutrients lead to food secure communities and also improved human health. So that's the link. It is really an interesting coordination in that. Thank you so much for listening. Someone here, thank you.
Thanks, everyone. Really appreciate having everyone here. All the types of questions. You know, we're a community of practice. We're constantly learning all the time, and we welcome input and ideas and experiences. And as I said, there's different ways. Our LinkedIn RD site where people are constantly sharing experiences and asking questions about what's worked, what's not worked. So this community is for all of us, and we welcome you. I know we're at the bottom of the hour here, so we're going to wrap. But just want to say thank you, Warren and David, for that excellent information. Abby and Tobias, thanks for your back end support there, and we look for you next time. Thanks so much, everyone.